In the emergency room, an acute ischemic stroke presents as a catastrophic electrical storm. As blood flow to a region of the brain stops, oxygen levels drop, and billions of neurons fire uncontrollably, manifesting as focal neurological deficits. While the clinical team stabilizes the patient, a blood sample is rushed to the hospital laboratory. Moments later, the clinical pathologist receives an automated Delta check alert. The interface displays a massive spike, plasma glutamate surging past 100 micromoles per liter, while ALT and AST enzymes spike above 1,000 units per liter. These specific numbers suggest a severe contradiction. A transaminase spike of this magnitude typically indicates acute hepatocellular necrosis, the physical rupture and death of the liver, yet the patient is currently in the midst of a localized brain crisis. The physician must determine if this data represents a multi-system organ collapse, a brain actively flooding the bloodstream with excitotoxins, or a pre-analytical illusion created during the blood draw. Understanding this anomaly requires looking at the anatomical context of glutamic acid. Depending on its physical location, the molecule shifts between a metabolic waste carrier and a high-speed neurological signal. Early life required a safe way to move organic nitrogen without exposing cells to toxic free ammonia. Evolution selected glutamate as the universal container for this traffic. Glutamate's formula is C5H9NO4. Operating as a dicarbolic anion at physiological pH, it carries a fixed negative charge at its tip. Compared to its shorter relative, aspartate, glutamate features an extra methylene group in its carbon chain. This extra link provides exceptional rotational flexibility. The negatively charged tail can rotate and bend to snap into complex protein locks across a wide range of molecular distances. This flexibility allows cells to bundle dangerous nitrogen into a standardized format. In the liver, specialized enzymes crack the molecule open, funneling the nitrogen into the urea cycle for safe clearance. This structural adaptability links the basic carbon metabolism used for energy directly to the global neurological drive of the central nervous system. A healthy adult stores up to 110 grams of glutamate. 65% is integrated into skeletal muscle, while the brain maintains a dense internal concentration. To prevent synaptic interference, the body suppresses circulating plasma levels, maintaining a steep 1,000-fold boundary. This strict boundary faces a dietary challenge from protein-dense foods and MSG, but this load never reaches the brain. The intestinal lining acts as a metabolic bouncer.
Enterocytes extract and consume 80% of dietary glutamate on the first pass, burning it for structural integrity. Any remaining glutamate that enters the blood is met by the blood-brain barrier. Capillary endothelial cells use active transport pumps to forcefully eject rogue glutamate from the brain's environment back into the bloodstream. Because the gut consumes the majority of the intake and the brain actively pumps out the rest, systemic neurotoxicity within the sealed environment of the central nervous system, glutamate is the master excitatory neurotransmitter. It facilitates more than 90% of all fast synaptic transmissions. During memory formation, a neuron releases glutamate to open NMDA receptors, triggering a controlled influx of calcium. This signal instructs the cell to build more receptors, physically hardwiring the memory through long-term potentiation. To prevent overstimulation, helper cells called astrocytes use EAA2 transporters to act as high-energy vacuums. They constantly sweep the synaptic fluid to keep ambient glutamate levels below 1 micromole per liter. An acute ischemic stroke cuts off the oxygen and ATP energy required to run these astrocyte vacuums. The pumps fail instantly. Without active clearance, synaptic glutamate levels surge from less than 1 micromole to over 100 micromoles per liter.
This massive surge jams the NMDA receptors open, causing an uncontrolled tsunami of calcium to flood the neuron. The calcium flood hyperactivates digestive enzymes that dismantle the cell from within. The neuron effectively fires itself to death, undergoing rapid autolytic necrosis. In a stroke, the surge of synaptic glutamate kills more neurons than the initial lack of blood. The widespread destruction is a structural containment failure of the body's own memory molecules. The efficiency of this glutamate clearance system changes significantly across the human lifespan. Preterm infants have lower baseline expression of astrocytic EAAT2 transporters and clearing enzymes. This immature clearing architecture makes the neonatal brain prone to excitotoxic injury during even minor hypoxic events. In the geriatric brain, the natural aging process causes a slow, measurable reduction in the number of EAAT2 vacuum pumps available to clear the synapses. This loss of suction creates a chronic, low-grade excitotoxic stress. Over decades, ambient glutamate mildly overstimulates the neurons, damaging dendritic trees and contributing to the progression of Alzheimer's disease. The preservation of human cognitive function depends on the lifelong mechanical efficiency of these microscopic astrocytic garbage collectors. Determining if a patient's brain is flooding with glutamate requires measuring plasma levels in the laboratory. The gold standard for this measurement is liquid chromatography tandem mass spectrometry, or LC-MS-MS. The machine vaporizes the plasma, using an electromagnetic grid to isolate particles with a mass-to-charge ratio of 148.1. To differentiate isomers, the isolated gas enters a collision cell and physically shatters. The unique daughter fragments at 102.1 provide exact nanomolar specificity. Despite this precision, the machine is blind to pre-analytical human errors that happen before the blood ever reaches the lab. If a blood tube sits at room temperature, background enzymes convert glutamine directly into glutamate. The blood secretly manufactures a fake glutamate spike while it sits on the counter. Mechanical hemolysis is even more common. If a phlebotomist pulls the needle too hard, vacuum pressure physically pops red blood cells inside the tube. Red blood cells contain significantly higher concentrations of glutamate and transaminase enzymes than plasma. When they burst, they dump their internal cargo into the sample. In our stroke patient, the transaminase alert and the glutamate spike were purely chemical artifacts. A rough needle draw ruptured the red blood cells, creating a profile mimicking acute liver necrosis. Preventing these illusions requires blood to be drawn after a 12-hour fast, placed immediately on crushed ice, and centrifuged within 30 minutes to separate cells from plasma. Laboratory quality control must maintain an allowable error margin of plus or minus 15% and a reference change value of 14.2% to differentiate metabolic shifts from analytical noise. Diagnostic stewardship dictates that these panels be reserved for tracking acute ischemic injury or investigating metabolic defects in infants. Routine ordering for generalized fatigue drives unnecessary costs and increases the risk of false alerts. A key interpretive pearl is to distinguish the free amino acid substrate, glutamate, from the structural enzymes ALT and AST. One tracks metabolic transport, the other indicates physical tissue death. Glutamate's architecture allows it to function as the primary nitrogen hub, fuel for the intestinal barrier, and master trigger for the central nervous system. This single flexible molecule links the basic metabolic processes of the gut directly to the highest cognitive functions and memories of the brain. Clinical intuition requires the active integration of two distinct fields, the raw biology of the suffering cell and the exact physics of the laboratory measurement.